Hello, my dear student. Welcome to Biology. Today, lecture 5, part 11. So, my dear student, we are learning post-fertilization structures and event in flowering plant. And last class, we have discussed about development of embryo, an important event in post-fertilization. So, we came to know about development of embryo in monocotyledon as well as in dicotyledon. We came to know that how from zygote have developed into embryo. Okay, my dear student, today we are going to learn a very important process of post fertilization that takes place in flowering plant, that is the formation and development of seed. So let's learn about development of seed. So my dear student, we know that seed is the end product of sexual reproduction in flowering plant. The seed is the final product of sexual reproduction in angiosperm. So seed is the final or the end product of sexual reproduction in flowering plant. And we have learned about various events in sexual reproduction, and this is the end product of sexual reproduction. So this is the aim of each and every sexual reproduction showing flowering plants, that their aim is to produce seed. And now we are going to learn that how seed is developed in flowering plant. Okay, now we came to know that seed is the final and the end product of sexual reproduction. Seeds are often described as fertilized ovules. So seeds are often described, or it is often explained as fertilized ovules. So these are ultimately fertilized ovules and are formed inside fruits, and they are formed always inside the fruits. Okay, now look at the diagram. Here a flower is shown. Okay, and from different parts of the flower, how the fruit or the seed formation have taken place here. Can you see? So okay, we have learned some of the parts of the flower. Yes or no? Okay, already we have learned about the parts of a flower. Okay, now can you see here the triploid endosperm cell? Okay, so triploid endosperm cell, which was formed during double fertilization. Okay, so the product of double fertilization was triploid endosperm cell and zygote. Now what happened after fertilization? What happened? This triploid endosperm cell has formed what? Endosperm. Now you can see the endosperm where it is present. Can you see it? So endosperm is present inside the seed. It is leveled here. Okay, then again you see the zygote. So zygote develop into embryo. Okay, and can you see where is the embryo? Embryo is within the seed. Now endosperm and embryo, okay, these two are inside the seed. Now can you see the integument? Okay, outer integument, inner integument. Okay, the external, okay, covering of ovule already we have learned. Can you remember the embryo sac was protected with more than one layer that are known as integument, and that integument is developed into seed coat. Now can you see the seed coat? Okay, now seed is fertilized ovule. And what is fertilized ovule? Embryo plus endosperm plus seed coat. Okay, so we can say what is seed? embryo plus endosperm plus seed coat. So these three together is known as seed. Now can you see the ovary develop into fresh fruit. So this is the fresh fruit part. So it develops into fresh fruit. Now these are the different parts of the flower which on time develops into fruits and seed. Okay, so what is seed? Seed is the final product of sexual reproduction in flowering plant, and seed are fertilized ovule, and these are formed of embryo, endosperm and seed coat. Clear? Okay. Next, here is the diagram. Look at the diagram. So a seed typically consists of seed coat, then cotyledon and embryo axis. So a seed typically consists of seed coat. Okay, so here one diagram is meant for monocot, that is corn seed, and another is dicot, that is bean seed. Okay, bean seed is an example of dicot, whereas corn seed is an example of monocot. So here both the diagram you can see the seed coat. Have you seen the seed coat? So it is labeled here, and for dicot the seed coat has two layers. Can you see? Testa and tegmen. So these two layers are there, testa and tegmen, and these two together is known as seed coat. Now what is the function of seed coat? The function of seed coat is to protect the seed. Okay, so seed coat protects the seed. Another is cotyledon. Now can you see the cotyledon here? Okay, so cotyledon, monocot has one only single cotyledon, whereas dicot has two cotyledon. Okay, then embryo axis, that is epicotyl, hypocotyl, radial. So all these are together we can say these are embryo. So embryo develops into a new plant. So you can see this part of embryo, that is inside the seed. Can you see this embryo? So this embryo in future develops into a new plant. Now can you see the important, okay, that embryo is protected by the seed coat so that no harms, no external factors affect them, and before the development of a new plant this should not be destroyed. Okay, so embryo in future develops into a new plant. Now can you see that in monocot that endosperm stores reserve food, whereas in dicot what happened? They almost used all the that endosperm reserve food for the development of embryo. Okay, so these are the two diagram that shows monocot and dicot seed. I hope that you have understood. So a seed typically consists of seed coat. What is the function of seed coat? To protect the seed. Then cotyledon are there. In monocot one cotyledon, whereas in dicot two cotyledon. Then embryo axis or embryo. You may see they are labeled here. Okay. Now seeds are of two types. So seeds are mainly of two types. Number one, endospermic seeds or albuminous seeds. So what do you mean by endospermic seeds or albuminous seeds? So albuminous seeds that retain a part of endosperm as it is not completely used up during embryo development. So what is albuminous seed or endospermic seed? In that seed what happened? A part of endosperm remain and it is not completely used up during the development of embryo. For example, wheat, maize, sunflower. So here a diagram is shown. Okay, endospermic, you may see this is a seed, that is endospermic or albuminous seed. So you may see here that this is the part. Okay, so here the some part of endosperm remain here without utilization during the embryo development, and because of which this type of seed are known as albuminous seed, okay, or endospermic seed. Some examples are wheat, maize and sunflower. Another type of seeds are non-endospermic seeds or non-albuminous seed, which have no residual endosperm as it is completely consumed in embryo development. So for example, pea groundnut. So in case of second diagram, you may see non-endospermic seeds. So in non-endospermic seed, you may see storage cotyledon. So cotyledon is the storage. Here you will not find endosperm. Okay, so no endosperm remains here, as during the embryo development all the food that is retained in endosperm or that remain in endosperm are consumed during the embryo development. And some example are pea and groundnut. Okay, so we may say that pea and groundnut, these are non-albuminous seeds, whereas wheat, maize, sunflower, these are albuminous seed. So today we have learned about two type of seed: endospermic seed or albuminous seed. These seeds contain a part of endosperm. Okay, because during embryo development these endosperm are not completely used up. Okay, whereas in non-endospermic seeds or non-albuminous seeds, there is no endosperm. Okay, why? Because these endosperm are completely consumed during embryo development, and example are pea and groundnut. Okay, so today we came to know about the development of seed, how seed is developed. Okay, so what are seed? Seed are fertilized ovule. Okay, and these seeds are composed of embryo, then endosperm and seed coat. Okay, so I hope that you have understood the development and formation of seed, then two type of seed. Okay, so thank you.